Really nicely introduced me. I'm Helena Hartmann, and I'm very, very honored that I'm able to present you parts of my research that I did for my PhD, or am still doing for my PhD today. And Alison already told you a lot about me, but I still have a slide about an introduction, so I'm just going to show it to you anyway. I did my master in clinical and biological psychology with Georgia Silani at the University of Vienna. And here I first came into the contact with neuroscience combined with psychology, because I investigated how individuals with autism spectrum disorder process the emotions of other people and what influence their own emotions have in this regard. And this was in the domain of social pain. And then for my PhD, I switched to the lab of Klaus Lamm, trying to investigate empathy and pro-social decision-making in the domain of physical pain. And next year, I will also extend this and hopefully finish my PhD at the Social Brain Lab in Amsterdam with Christian Kaisers and Valeria Gazzola. And then part-time, I work as a scientist and psychologist for My Mind, which is a startup company in Vienna that develops Brain Hero, as Alison already said, that is a neurofeedback game for individuals with autism spectrum disorder. So my research combines social, cognitive, and affective neuroscience, which is funnily also the name of the lab that I'm working at, with a psychological background. So I'm very interested in the brain during the perception and processing of different types of pain and the modulation of pain by placebos. So treatments that don't have any real effect on a pharmacological level. But I'm not only fascinated by how we feel pain ourselves, but how we perceive the pain of others around us and whether this changes our behavior towards others. And this overlap that you see in the middle, this is what my PhD is all about. So how are one's own pain processing and someone else's pain related? Is there some kind of shared mechanism between the two? So if we try to define empathy in simple words, we can say it's the sharing as well as understanding another person's emotion. For example, you see your friend hitting his or her elbow on the door frame, and you kind of immediately feel what the other person, so what your friend feels. Of course, in our lab, we have a bit of a more complex working definition that defines empathy as a process consisting of multiple different components. So first, we have to either directly observe or imagine the emotions of another person. Then we have to be in a state of isomorphic affect sharing, meaning that we feel the same as the other person. This is also called feeling with the other person. And then thirdly, we need some kind of self-other distinction mechanisms. This means that we have to know the source of the emotion we are feeling. We have to know it's coming from the other person and not from ourselves. So empathy is a combination of more automatic bottom-up, but also more controlled top-down mechanisms. And I focus on this affect sharing component in my PhD, and I want to clarify the relationship between self versus other directed emotional experiences in the domain of pain by investigating how these processes are implemented in the brain. So this definition also differentiates empathy from other mechanisms, for example, emotional contagion, where we are automatically feeling something similar as another person, like contagious yawning is a similar concept, if you know that. We can also differentiate from personal distress, where we feel the emotions of another person, but we are missing this self-other distinction. So we are really strongly, mostly negatively affected by the emotions of other people. Then we have concepts such as sympathy or compassion, where instead of feeling with the other person, like in empathy, we feel for the other person. And then what follows is pro-social behavior, where we then actually help another person. So there are many reasons why feeling empathy and investigating how it is processed in the brain are important. First of all, we are inherently social beings, so be it in friendships, at work, or in romantic relationships, we constantly have to process and understand our surroundings. And a big part of these surroundings are interacting with other people and dealing with emotions that are kind of flying around. So these emotions can come from other people, and they can come from ourselves. They can be both positive or negative. And empathy can help us process these emotions, respond appropriately in social situations, and it can also help to foster bonding. The majority of social neuroscience research has used somatic pain as a model to investigate empathy. And there are two very good reasons. I'm sure there are more. But first of all, pain is something that can be very easily and robustly be induced in psychological experiments. So if I give someone a painful shock, he or she will probably feel it as painful every time, which is good for us as researchers. And second of all, there's already very strong evidence regarding the neural networks underlying or involved in pain processing. But before I explain the two major components of the pain processing network in more detail, I want to give you a quick explanation of how pain is defined. It constitutes an unpleasant sensory and affective experience associated with actual or potential tissue damage. And as you may have noticed, this definition is not necessarily limited to somatic first-hand pain, so pain we feel ourselves, but it could also include other types of pain involving other people, like social exclusion or just seeing others getting hurt. So just as a short, let's say, internal exercise for all of you, I'm going to show you 3 pictures. I don't need to know any reactions now, but just maybe observe in yourself what your initial reaction to these pictures is. And I would be interested to hear your experiences later or how you perceived the pain of the people in the pictures that I'm about to show you. And then we can also discuss it. So we have the oven. We have a weight. And we have a shoe. So maybe this hurt yourself, maybe this didn't hurt at all. I think that empathy is something that is very individually different, and everybody kind of feels it in a completely different way. That's why it's so interesting, I think. So if we look at firsthand pain, so pain we feel ourselves, pain is something that it's considered to be a complex, multidimensional experience. And usually it's divided in 2 major components. One is the sensory-discriminative aspect. This one is a more basic representation of the pain, for example, the intensity, the location, the timing, or physical characteristics of that pain. And it's usually represented in the primary and secondary somatosensory cortices in green. And then there's the affective-motivational component of pain, which describes more the subjective judgment, so the unpleasantness we feel and the intention of withdrawing from that pain. And this is coded for in the brain, for example, in anterior and mid-cingulate cortices in red and anterior insula in brown. So if we now look at how people process the pain of others, there are of course lots of differences, but also some similarities. So in green, you see the first-hand experience of pain and what is active in the brain when we've experienced pain ourselves. And in red, you see the overlap between first-hand and empathy for pain. So what is common between the 2 processes? And in fact, it has been shown many times in the past that receiving pain and observing others in pain recruits overlapping activation in these 2 networks that I just presented, so somatosensory and affective network. And mostly this overlap occurs in regions coding for the affective component, but some studies also showed an overlap in somatosensory areas, and they highlight the importance of this component. And this research has been taken to suggest that shared neural representations may underlie both of these experiences. This means that we might recruit the same mechanisms when we feel pain ourselves and when we see someone else in pain. But it is unclear whether and how we can infer that shared activations, so brain activity that we measure, for example, with fMRI, are equal to shared representations, so the underlying functions or mechanisms. So if we actually recruit the same mental processes during empathy as when we directly experience the emotion we're empathizing with. And this gap has recently been addressed by the so-called placebo empathy analgesia studies led by Markus Rüttgen from my lab. And they employed a more causal approach by manipulating the first-hand experience of pain using placebo analgesia. And then they looked whether this also had an effect on empathy for pain. So just to show you quickly what they did, half of the subjects were led to believe that they received a pill that had a strong analgesic effect and that this pill would reduce their own pain. In fact, this pill was a placebo, so it had no pharmacological ingredients whatsoever. And then there was also a control group who did not receive any pill. And then participants went into the MRI scanner and did a task where they either received electrical stimulation on their hand, or they observed another person sitting outside the scanner also receiving such stimulation. And if we look at the results, this is for first-hand pain, so when participants received shocks themselves. The placebo was able to reduce the self-perception of pain, which is not surprising, but this is still nice to see and to check. So in dark gray, you see the control group ratings and the brain on the left side. And in light gray, you see the ratings of the placebo group. And then in the figure on the right, you see brain activity. And we also see that in bilateral anterior insula and AMCC, brain activity to one's own pain was reduced in the placebo group compared to the control group. But what's even more interesting is that the placebo manipulation also had an effect on empathy. So if we look at the behavioral ratings, the authors found reduced subjective empathic pain and unpleasantness. So they asked 2 questions related to empathy. The first one was, how painful is it for the other person? And the second one was, how unpleasant do you feel? And again, we see the same pattern. So in dark gray, the control group, and in light gray, the placebo group. And now that we look on the brain, it's even more interesting because again, activity in these 3 regions, bilateral anterior insula and AMCC, was also reduced in the placebo group when the other person received a shock. So as a follow-up, the authors wanted to investigate even more the neurochemical mechanism behind this effect by directly interfering with it. And so here they targeted the endogenous opioid system because it plays an essential role in pain regulation, including placebo analgesia, and because regions like the mid-cingulate cortex have a high density of opioid receptors, and in general, regions of affective pain processing. So the setup of the second study differed only in the sense that the participants got a second pill after the administration of the placebo, which was either also a placebo or it contained naltrexone. Naltrexone is an opioid antagonist that is able to block opioidergic pathways. And interestingly, well, then they did the same task with electrical pain, the participants getting shocks themselves and another person getting shocks. And what they found here is that naltrexone was able to reverse the effects of placebo analgesia and kind of normalize empathy levels back to levels without the placebo. And this suggests that self-pain and vicarious pain, so empathy for pain, may indeed be grounded in similar neural and neurochemical processes, or in other words, shared representations. However, to sum it up a bit, some of the empathy studies in the past found an overlap in activation in the affective-motivational component of pain. Others found involvement of sensory-motor or somatosensory areas. Placebo analgesia in the study that I just presented only reduced brain activity in the affective component of pain, so insula and AMCC. Although the manipulation, so placebo analgesia, generally also affects the sensory-discriminative component if we look at first-hand pain. So this mismatch might have resulted from the choice of paradigms that the authors had employed in their study. And here, empathy research often makes a distinction between so-called cue-based versus picture-based tasks. So on the top right, you see these symbols like the arrow and the lightning and the facial expression. These are so-called cue-based tasks where you have to infer the pain of the other person via abstract symbols. Usually this is accompanied by getting some kind of physical pain like electrical stimulation. On the other hand, there are picture-based paradigms that you see on the bottom right, where the stimulus material is a bit more visually explicit. So here we usually see body parts like hands or feet in very painful situations. These are also like the pictures that you saw before. And this could lead to the hypothesis that somatosensory components might only be involved when empathy is instigated by really strong visual cues. So when the attention is directed to the location or the type of pain. And in fact, most of the studies in the past that looked at an overlap between firsthand and empathy for pain usually find a role of somatosensory pain processing networks when they employed picture-based tasks. And the previous placebo analgesia studies, they did not use such a picture-based setup because they used a cue-based task. So therefore, in this pre-registered study, we wanted to know whether placebo analgesia can also interfere with the sensory discriminative component of empathy for pain, and what role in general somatosensory representations play in empathy. So our hypothesis, similarly to the placebo analgesia studies that I presented, was that we would observe behavioral and neural reductions in empathy, but that those reductions would be restricted to the specific location where placebo analgesia was induced. So in order to make this study tailored to somatosensory pain processing, as I will explain in a minute, we induced the placebo not in a general way via a pill, but on a very specific body part. So here's an overview of the session. Participants came to the MRI scanner together with another person who was posing as a second participant, but who was in reality a confederate of the experimenters. We conducted a pain calibration and a placebo analgesia induction that I will explain in more detail on the next slide. And then participants completed 2 empathy for pain tasks, specifically tailored to observing a possible involvement of the somatosensory component. So we conducted this pain calibration individually for the right and left hand because it has been shown in the past that pain thresholds can vary between the 2 hands, even within a person. And we gathered average pain ratings for high pain, medium pain, and low pain. We then had a study doctor coming in and doing the placebo induction, where the participants were told that a strong local anesthetic would, in the form of a gel, would be applied on the back of their right hand, while the left hand would only receive a basic skin cream. In reality, both of those gels were nearly identical and they had no active pharmacological ingredients, so it was a placebo on the right side. And then we also conducted a classic conditioning procedure to amplify effects of this placebo even more. And here what we did is we had electrodes attached to the backs of the hand, and then we told participants we would give them high pain stimulation that we had calibrated before on each hand. And on the left hand, this is also what we did. We gave them high pain and participants really felt that. So this was good. But then on the right hand, we secretly lowered the stimulation intensity to medium pain. So participants thought they were getting high pain stimuli, but on the right hand, it kind of felt a bit less painful than on the left hand. So this was there to suggest substantial pain relief by the gel. And this was just as a boost, so to say. This is sometimes used in placebo analgesia studies, but it also doesn't need to be there. So sometimes the suggestions by the doctor are enough to induce a placebo effect. And then participants went in the scanner. So just to show you a bit also, so you can imagine a bit better what the setup looked like at the scanner where we did the study. So here's me posing as a study doctor just for display purposes, explaining the medication to the participant. And this is kind of what the electrode setup looked like. So we had 4 electrodes per participant, 2 on each hand, 1 for painful stimulation and 1 for non-painful stimulation as a control. And we had a lot of cables in the scanner. It was really crazy. And then in the scanner, it looked like this. So the participant was, of course, lying inside the scanner. And then next to the scanner, we had a table with a fake computer set up where the confederate sat. And of course, the confederate also had electrodes going from his or her hands outside to the pain stimulator. So it seemed realistic. We had some criteria to identify non-responders to the placebo manipulation. This is usually done because not everybody responds to a placebo in the same way. So we oriented ourselves on the previous study by Markus Rüttgen and colleagues and specified some criteria. And those were, for example, if participants had strong doubts about the whole cover story. So if they told me during the session, I think I got a placebo, I still feel very high pain, I think the doctor wasn't real, things like that. We also excluded participants who needed a very high number of conditioning trials in order to feel this relief on the right hand. So in order to believe what we were trying to tell them. And then we also measured belief in the medication effectiveness. And if this belief was low in general, or it kind of decreased over the session, we excluded people and we classified them as non-responders. And then finally, because our study was a within subjects design, we could also directly compare the first-hand pain ratings of people, because our first task involved also getting pain firsthand. So if the stimuli that were given on the placebo hand, so the right hand, were rated as higher or similar to the pain that they got on the left hand, we would classify them as non-responders as well. So in order to, let's say, move on to be part of the analysis or part of the final sample, they needed to fulfill all of these criteria. So of course, I tested a lot of people, and in the end, I had 45 subjects who showed a first-hand placebo effect. This was specified in our pre-registration, where we said that we would continue the data collection until we had reached 45 responders. And half of those were male, and they were about 24 years old. And importantly, all of those participants were strongly right-handed. This is the first task that we did. So here, participants received either painful or non-painful electrical stimulation themselves on each hand, or they observed the second person receiving such stimulation. So this task is very similar to the one by Markus Rüttgen and colleagues in the placebo empathy analgesia studies. The only difference being is that we tried to make it a bit more somatosensory focused by showing pictures of either the participants' hands from an egocentric perspective or the hands of the confederate to kind of draw the attention to the location of pain. And we also had the cues appear on the hand to show whether, for example, if a red circle appeared, this would be a painful shock on the right or left hand, depending on where it appeared. And then, of course, they also received the shocks, or the other person receives the shocks. And then they rated the pain and unpleasantness that they felt. We had 2 manipulation checks, even though we had already excluded non-responders, just to be really, really sure that our placebo induction had worked. So 3 times during the session, as I already mentioned, I asked participants how effective they thought that the pain gel on their right hand would be able to reduce their pain in this location later in the task. And I asked this right after the gel was applied by the doctor, preconditioning, then post-conditioning right after the conditioning procedure, and again after all tasks were completed post-session and participants came out of the scanner. And what we found here is a significant increase in belief from pre to post-conditioning, so from the first bar to the middle bar, showing that the conditioning procedure had some kind of effect on the subjective belief in the effectiveness of this medication. We also observed a significant decrease until the end of the session, so from the middle bar to the right bar. However, this decrease did not drop lower than the initial belief. And as you see here, the scale is from 0 to 10, and the belief was already quite high to begin with on average. So we still took this as enough evidence that the participants had a placebo effect that was strongest in the middle of the session and only decreased slightly. And important to know, all of the tasks were done after the conditioning procedure, so when the belief in the placebo was highest. As a second manipulation check, I also asked participants after the session how painful on average the stimulation felt on the right hand with the medical gel and on the left hand with the control cream, or the basic skin cream. And here we also found a significant difference between the right and the left hand. So in orange, you will always now see the placebo hand, and in blue, always the control hand. And of course, these two manipulation checks are, let's say, a little less surprising because we did already include only responders to the manipulation to these 4 criteria that I just showed you. So let's have a look at the behavioral results of this first task. As we can see here, the electrical stimulation participants received in the task on their placebo hand in orange was rated significantly less painful than the stimulation on the control hand shown in blue. So this is again a nice manipulation check, but again, we did already separate out the non-responders, which this is why this rating makes sense here. But this was nice because our prerequisite in order to investigate effects on empathy was to have a significant or a very strong first-hand placebo effect. However, if we look at the 2 empathy ratings, we see that this effect did not transfer to empathy, where the pain and unpleasantness ratings were comparable for the 2 hands. And this was the case for both of the empathy questions that we asked. So we had asked how painful was it for the other person, and how unpleasant was it for you to observe the other person getting a shock. In both of these, participants rated the pain of the other and their own unpleasantness similar for the 2 hands. So our initial conclusion was, okay, there is no influence of a location-specific placebo induction on empathy. These are again the same results for first-hand pain and empathy for pain that I just showed you. However, if we look again at the first-hand and empathy for pain ratings, we observed that empathy ratings for both hands seem to be closer to the ratings of the placebo hand if we look at first-hand pain. So this was a bit puzzling to us. So we tried to compare it with the previous study by Markus Rüttgen and colleagues. And as you can see here, we don't see the same pattern because the placebo condition alone showed the decreased ratings, and both the control conditions were higher. So of course, this is highly preliminary and we didn't directly investigate this, so it is a rather exploratory finding. And a big difference between these 2 studies is also that ours was within subjects, so we induced the placebo within the participants, and the other one was between subjects. But if we want, we could speculate that our placebo induction maybe did have an effect after all on participants, but in a more generalized way instead of a localized one. And I'd be happy to hear more theories from you later, what this could mean or what other explanations there are for this. We then conducted region of interest analysis in primary and secondary somatosensory cortex, as well as in the 3 regions that had already been found in the previous study, bilateral anterior insula and AMCC. Today, I will focus mainly on the somatosensory brain regions, as this was our main research question. However, we did not find any modulation by the placebo manipulation in bilateral S1, neither for self nor for other, so empathy. In S2, we did observe evidence for a first-hand placebo analgesia effect in right S2. Here, activity corresponding to the right placebo hand was lower compared to the left control hand. But again, we found no transfer of this effect to empathy. So to sum up the results for task 1, we did find evidence for a strong localized placebo effect during first-hand pain in behavior and in right S2. But empathy for another person was not influenced by a localized pain reduction in a specific body part. And we also did not find evidence for somatosensory sharing, so a transfer of this to empathy in S1 or S2. Maybe, possibly, we found a generalized effect of the placebo on empathy for pain ratings, but this will have to be investigated further in the future. For the second task, we then made the stimulus material even more explicit and focused more strongly on showing the location of the pain. And these are also the stimuli that you had, that I showed you before. So here we had stimuli showing everyday painful situations. For example, a hand being burned on the stove, cut while gardening, or hit with a hammer. Importantly, the location of pain was always the back of the hand. And to be honest, this was actually quite difficult to find enough situations where the pain was happening on the back of the hand, because if you think about it, it doesn't always do. So sometimes it's the finger, sometimes it's the arm, but we made it work. We found 15 situations that were very painful for people. And we did a validation study and validated those. And then in this task, participants just saw the picture, and then they again had to rate the pain of the person shown in the picture, and then also their own unpleasantness. And importantly, this was a 2x2 design. So participants saw pictures where a hand was in pain or not in pain, or they saw pictures where they had to rate the left or the right hand. And the hand that they had to rate was always marked with a black arrow. And here is an example of me shooting the stimuli. So this is me with my hand in a paper cutting machine. And this was actually the most difficult picture to take, because I had to simultaneously try to make it look realistic and also hope to not get my hand cut by the machine. And in the end, sadly, this picture didn't even make it in the final stimuli set because it was rated too unrealistic. So if we look at the results of this task, again, we have the 2 empathy questions that we had. We did not find a transfer of the placebo effect to empathy for everyday painful situations. So again, the ratings for the right and left hands in the pictures were similar. So again, it seems like the placebo didn't influence the ratings, right? But we were still curious to see whether we could also, again, find evidence for a more generalized effect of the placebo on empathy. So luckily, we had done this validation study where the same pictures had also been rated by an independent sample of 38 people. Here you see again the ratings from this task, or from the study that I presented. So we also had a look at the unpleasantness ratings in the validation study and compared this to the ratings of our main study. And indeed, unpleasantness in the main study that you can see in pink here was rated in general significantly lower than unpleasantness in the validation study. And of course, this can have many reasons because they were also not completely identical. And this analysis is still very preliminary, but I wanted to put it in to just show you the full picture. So I'm happy to reason with you what this could mean. And this will have to be investigated more specifically in a future study because, again, it was not part of our pre-registered predictions. Also for this task, we conducted the same region of interest analysis in the same regions. And again, we found no effect of the placebo on bilateral S1 and bilateral S2. And here we even did additional post-hoc Bayesian analysis to really confirm that there was nothing, and there really was more evidence for the null hypothesis than the alternative hypothesis. Interestingly, in the 3 affective regions, so anterior insula and anterior mid-cingulate cortex, we found evidence in bilateral insula for the opposite effect. So for an increase in brain activity for pictures relating to the right placebo hand. So this is the opposite of what we had expected. And we were really quite puzzled by this result. And this will definitely have to be followed up. For example, it could be that because we tested only right-handed participants, the right hand was maybe in general more salient and participants paid more attention to it. This is just one explanation. So to sum up the results for task 2, again, placebo analgesia did not reduce picture-based empathy in a location-specific way for the behavior. And also, we did not find a reduction of activity in affective and somatosensory brain regions in this location-specific way that we had hypothesized. We did find increased brain activity in bilateral insula for stimuli corresponding to one's own placebo hand, which was quite surprising. And we might also have found evidence for a more generalized effect of the placebo, but this time on the unpleasantness ratings. So to conclude, Empathy for pain may thus rely more on sharing another person's affective compared to their somatosensory specific state. So the general processing of another's pain might have a higher weight than the processing of the exact location or source of that pain. And if we think about it, to me at least, it makes sense that if we see someone in pain, the first thing that's important is this person is in pain and we share that pain. And maybe only in some very specific situations, like in hospital or emergencies, it's important to also additionally know the location and the source of the pain. So for example, whether the knife is in the heart or in the leg. This research also showed that it's very important to consider the task that you choose when you investigate empathy, because there have been quite some differences found in different empathy tasks, reminding you again of cue versus picture-based. So in total and in general with my PhD, I hope to refine the understanding of the neural underpinnings of empathy, which is very important when we try to characterize the magnitude of influence that our own pain experience has on empathy. And of course, I want to thank my lab and my 3 co-authors, Federica Riva, Markus Rüttgen, and Klaus Lamm, my supervisor. And I want to thank you for your attention, and I'd be happy to answer any questions or comments. Thank you. I mean, you can also write the questions in the chat if you want. I guess it's a good sign if there are no questions. What do you think, Alison?
We'll get a couple. Yeah, so actually I just have like a clarification question, and it's related to when you're comparing the like placebo and control for other perception to like the placebo trial of the self condition. In task 1, I believe, with some of the past work. And I was wondering if it was like expected, or I think and I could be misremembering at this point. It's been a few slides away, but I noticed that for all of the self conditions in that past work, in all of the other conditions of the past work, the magnitude of the activity is lower than for in the self condition, even lower than the placebo. And so, yeah, I'm wondering, is that like a common finding? Like, should we just expect that we just don't have as much activation to others' pain in general, or is there a different kind of explanation?
Yeah, so that is usually the case, that of course first-hand pain is something very salient. We feel it ourselves, so we usually get stronger activation to that if we compare it to empathy. But I think the last study, so the study by Markus Rüttgen about placebo analgesia, they also found comparable levels of pain in the self and the empathy conditions. So they did the direct test between the two, let's say the ratings and also the brain activity, and they did find that it was comparable, and also that the effect sizes of the differences between control and placebo conditions were comparable. And usually what is also the case, because in empathy we ask how painful does it feel for the other, but also how unpleasant. And this is trying to tackle two separate aspects of empathy. So asking about the pain of the other is more the cognitive evaluative aspect, while the unpleasantness is related more to this effect sharing component. And usually if we just have to rate the pain intensity of another person, we are a bit higher than if we have to rate the unpleasantness. So the unpleasantness ratings are usually a bit lower than the empathy for pain ratings, because these are also just two very different concepts.
I feel like that's interesting. I was wondering what those unpleasantness scores were getting at, and I feel like that's especially interesting considering now that now that you've shown that like this somatosensory component of pain may not be how we're connecting to others, and that this this lower rating of unpleasantness or this affective component. Yeah, I wonder like under what conditions you can get that to spike, and if you can kind of like show the opposite to be true.
Yeah, no, definitely. I think it's very interesting.
I've got a question. I've got two questions for you. One being from the previous studies. Why only?
Can you speak up a bit because I can?
Yeah, sorry. I've got two questions. One about the previous studies. So you mentioned that you only used opiate antagonists, and I'm wondering why only that? Like, do you expect there to be similar analgesic effects with analgesia using other mechanisms? Or is there something specific to the opiate antagonists that is why it's working?
So I think I didn't mention it very elaboratively before, but the opioid system is only one of the neurotransmitter systems involved in these kind of processes. So of course there are many options, but I guess they had to start somewhere. And if we try to think of one that is maybe the most promising, I think that the opioid system is a great candidate because it has a lot of receptors in these areas of affective pain processing, and endogenous opiates are also released when we feel pain in order to lead like to an endogenous analgesic effect for us. So I think that was the reasoning of the authors in the past. But of course, they also write in their paper that, for example, the dopamine system can also play a very big role. And I think there is even a review that's going to come out soon from our lab that is talking about these different types of neurotransmitter systems and their role in empathy.
Second question is, one of your slides you talked about how people were rated, were rating the placebo effects from when they were given placebo, in the middle, and at the end, correct?
Mm-hmm.
Yeah. I'm just wondering, have you looked at your results in using the rate of decline of the effectiveness from post to the end of the study, as like to see why there might be such a big non-difference with what you're finding?
Yeah, I definitely tried that. I think I tried nearly everything. But so in this, and I also mainly presented the pre-registered results now and only some exploratory stuff, because of course not everything works that you try. And we didn't find relationships between those effectiveness ratings that were measured at 3 times during the session and the ratings or the brain activity. And I'm wondering why, because what we were trying to measure is kind of the subjective belief in the placebo, which should be related to the analgesic effects that you see in the ratings or in the brain. So I was also a bit surprised to not find any relation there. But what could be one explanation is that those ratings were measured quite separately from the task and from the actual, let's say, when they were in the scanner. So we always asked those questions while they were still sitting outside in the control room. So maybe this is why it was kept kind of separate. And what could also be is that we measured something completely different with the belief ratings than with what we then did in the task.
I have a question about the stimulus set.
Mm-hmm.
So, and if this is something you already talked about and I missed it, I apologize. But I was wondering about the decision to make the visual always from the first person perspective, because it seems to me like that would kind of work against the piece of empathy that you defined as being this distinction between self and other. It seems like it really strongly pushed people toward simulating the pain from a first person perspective, mm-hmm. rather than empathizing with someone else. So yeah, I was kind of curious about that decision.
Yeah, so it was very conscious. So we discussed a lot when we tried to think about the stimuli. And the first reason was that if we would have switched the perspective for the other person, This could have led to a confusion of right and left hand, just because participants have to think much more, okay, my right hand is here, but the participant's right hand is on the other side. So we wanted to be as easily understandable as possible. This is the first reason. And I was also afraid of having too much of an egocentric perspective. So I was afraid that participants would see the Confederates' hands and think like, those are my hands now. This is why I did include not the full picture of the hands on the screen, but I had like a black rectangle around it. So it was still, let's say, a more distanced picture that filled not all of the screen. So it was not like a direct extension of the participants' hands, but it was really a picture that they saw. So I think those were the main reasons. But indeed, there have been studies who kind of look at this difference between self-perspective and other perspective. So whether you empathize more when you try to imagine that you're this person, or whether you empathize more by trying to put yourselves in the shoes of the other person. And they do find differences between these two conditions. And usually the self condition leads to higher empathy just because you imagine it to be yourself. But this only underlines the relationship between self and other related processes, I think, because yeah, it's really easily, it's much more easy for someone to empathize if they relate in a personal way, I would say.
That makes sense. Thank you.
I have a question.
Yeah.
Is there any effect of like participant bias where the participants in the scanner and they see a picture of someone getting hurt, and then the experimenter says, how much pain are you in? And then they report that they're in pain less because they feel it, but more because they want to appease the experimenter.
Yeah, I mean, that's definitely a big problem in all psychology studies, I would say. Social desirability is something that we always have to take into account. And I did measure some questions about the expectations that participants had about the results of the study at the end. So I asked them what they thought that the placebo would do to themselves, what they thought that the placebo would do to their perception of the other person. And in fact, while most participants believed that the placebo or the medication, as they thought, would reduce their own pain, they didn't connect this to empathy, and they didn't think that this would also reduce empathy. So only very few participants said, well, this will also have an effect there. So I hope I kind of dodged the bullet with the social desirability, but yes, definitely, it's a big disadvantage. You can, of course, if you find questions later, you can also send me an email or follow me on Twitter. And then I'm happy to answer questions also later.
She has a great Twitter account if you're not following already. I love it. It brings me joy. I actually do have another question. This kind of goes back to one of the like first slides that you're presenting in your intro of like this idea of like the neural representation of pain. And I wonder if there was like, even though you're not seeing these like magnitude difference in these like univariate like approach, like I was wondering if there was plans to maybe look at this and like look at the pattern of representation instead and like see if there was distinct differences between these conditions and not just the magnitude of activity, but the pattern of activity within these regions.
Yeah, it's definitely on my to-do list. I haven't done multivariate analysis yet, but I do want to learn them anyway. So what better way to just use this data? Because I think this is something, of course, and especially since the studies of my lab, the previous studies, they found a modulation of empathy by the placebo. Of course, it was between subjects, but still I'm wondering whether I maybe missed something. So this will be definitely an idea for the future.